Hello everyone, this is Daniel Blackburn and this is another lecture of Environmental Soil Microbiology SWAE3411. Today's lecture is about sulfur cycle. First of all, a little bit about the sulfur. Sulfur is a very complex and highly reactive element. Atomic number is 16. The molecular weight of the most abundant isotope is 32. About 95% of the Earth's sulfur is S32, but we have also almost 5%, 4 point something percent of sulfur 34 also. There are many isotopes. There are about 30 isotopes from sulfur. Many of them are stable. For example, 33, 34, 36 are stable isotopes. So it makes it quite easy to study in isotopic fractionation of sulfur on different environments because of the availability of different stable isotopes, and also radioactive isotopes are available. The photograph here is about artisanal elemental sulfur mining from a sulfur volcanic deposit, volcanic crater. When we think about sulfur in the environment, because we have to think about sulfur as this dynamic molecule which possesses many oxidation states. So the oxidation states from sulfur can range from 6 to -2. And sulfate on the soil is the highly oxidized version of sulfur. It's the most abundant in soil environments, is in the form of sulfates and sulfate salt precipitates, like gypsum, for example. So sulfate is the oxidation state is 6, and it can be reduced to thiosulfate, elemental sulfur, and at the most reduced forms are sulfides and dimethyl sulfides. Normally, the fixation of the sulfate in organic molecules entails also the reduction of the sulfate into more reduced forms of sulfur. In the same time, when you have oxidizing condition, or aerobic conditions, you can have the opposite happening, and you have the oxidation of these molecules towards sulfates. So this cycling of these molecules in the environment is not about only the movement, but the type of a compound that can be moving through these environments depending on how oxidized or reduced these molecules are. Let's have a look on the biogeochemical cycle of this of sulfur. Yeah. So you can think about sulfur. Sulfur is very abundant on the Earth systems. It's about 0.1% of the Earth's crust, but it also has a very important atmospheric component. So in the atmosphere, you will have that sulfates and hydrogen sulfides will be the main forms of atmospheric sulfur, but also dimethyl sulfides also will be present from water emissions, yeah, from microbial emissions in water systems. These days also the contribution for atmospheric sulfur is coming quite a lot by the use of fossil fuels. A good portion of these fossil fuels contain sulfides, and the burning of these fossil fuels releases sulfates to the atmosphere. Normally, these sulfates, before the burn of fossil fuels, the sulfates would be coming from volcanic eruptions mainly to the atmosphere. Once you have a sulfate pollution on the atmosphere, the sulfate is dissolved in droplets of water and can come back into the soil, in the water systems, through acid rains. So when the sulfates dissolve in water, they become sulfuric acid, and the sulfuric acid is what we usually the main component of acid rain. So atmospheric pollution by burning of fossil fuels, before we have so many catalytic systems to filter this out. This was the main way in which the wet deposition of sulfates was happening on soils. Okay, dry deposition can happen by dust also, but the main form would be wet deposition in form of acid rain. But nowadays, atmospheric contamination with sulfate is low, quite low, so the acid rains have decreased, and we have now a problem where many environments are deficient in sulfur again, whereas before it was the opposite. They had plenty of sulfur because of the acid rain. So acid rain was an environmental problem, but at the same time was a source of an important nutrient for plants and microbes in many environments. So once these sulfate molecules reach soil environments, they can be immobilized in the organic matter and cycle back into sulfates, or they can be reduced and emitted back to the atmosphere as sulfides, or they can be used to make elemental sulfur and be stabilized as elemental sulfur in soils. Okay, so let's have a look on how it's naturally occurring sulfur in soils. In acid soils, you will have that the sulfur would be soluble in acid in the form of sulfuric acid, and the excess of sulfur in acid soils normally causes the dissolution of iron, and the solubility of iron will contaminate these waters with this red color that you see here. So this is acid sulfate soils are very, they can contaminate the environment through the loss of iron from these soils. But this is not the case from Oman. Normally what you have in Oman is the opposite. You have alkaline conditions, and these sulfate molecules will be tending to form precipitates with calcium mainly, as gypsum, but also other cations. And this is, of course, an extreme state where you have accumulation of gypsum salts in the desert of Palestine. In normal agricultural soils, of course, you will not have any of these extreme cases, but the main form of sulfur in the soils will be as sulfate and organic sulfur. Actually, the main form in soils will be organic sulfur. The most abundant would be organic molecules carrying sulfur, as it is with nitrogen. The reason being is because once it becomes sulfate, you can lose these molecules through different processes. So the main sources for agricultural systems of sulfate are atmospheric deposition, but not as much as we had before. Plant residues, animal manures, or organic matter that you can add to the soils, mineral fertilizers or amendments that contain sulfur. So once you add these amendments to the soil, they will tend to be oxidized and become part of the sulfate pool in the soil, and will be incorporated into the organic matter, into the microbial biomass, and will become part of the organic sulfur pool. And there's going to be a cycle between the inorganic sulfate and the organic sulfur pools. So whenever you have reducing conditions on the soil, or as to say anaerobic conditions, or waterlogging, for example, you will have that you're going to have a reduction of the sulfur into elemental sulfur and sulfides. Yeah, we have formation of elemental sulfur and sulfides. And whenever you have oxy conditions, you will have the oxidation of these reduced sulfur molecules. The main loss of sulfur from environments is through leaching and erosion, but also we have important emission of volatile compounds of reduced sulfide forms. Yeah? And one of the other main forms of sulfur loss from soils is through the biomass of crops that we harvest from the fields. All right, so let's talk a little bit about these individual processes that are happening in soil. First of all, between mineralization and immobilization, this is the interchange and the cycling between the sulfate in the soil and the uptake of the sulfate, the incorporation of the sulfate into organic molecules, and the mineralization will be the release of the sulfate back to the environment. This is an oversimplification. Some of these processes have to be cycled through other inorganic molecules, and there are steps in between this mineralization. And the immobilization often entails also that these sulfur molecules are being reduced to be incorporated into organic molecules. So when we look from the microbiology perspective, we can, in the same way we talked about the nitrogen cycle and the carbon cycle, we can look at the processes that are being aerobic, anaerobic, or can be both aerobic and anaerobic. So in this figure here on the left, we have the solid lines are aerobic reactions. The dashed lines are anaerobic reactions, and the dotted lines can be aerobic or anaerobic. So here in the middle you have assimilatory sulfate reduction when you incorporate the sulfate into the organic matter, and this organic matter can be completely reduced to hydrogen sulfide as part of anaerobic respiration or other processes. This is assimilatory because it renders organic matter. It's using the sulfate to build amino acids mainly on the soil, on these living beings. We have dissimilatory sulfate reduction, which is dissimilatory sulfate reduction is what we normally refer to when we talk about sulfate reducing microbes or sulfate reducing bacteria and archaea. This is part of anaerobic respiration, part of anaerobic respiration, and it uses sulfate as an acceptor of electrons in this process. We have disproportionation also. I will explain this a little bit more in a bit. And we in the oxic side of the cycle, you will have sulfide and elemental sulfur oxidation processes by colorless sulfur bacteria, and you will have elemental sulfur reduction by anaerobic microorganisms. Normally these are photosynthetic, like the arrows 4 and 5, which talked about the purple sulfur bacteria and green sulfur bacteria. These anaerobic microbes, they are photoautotrophs, but photolithoautotrophs. They use the sulfur as a source of electron also, and therefore they can be oxidizing this sulfur, but also they need sunlight for this photosynthetic mechanism. And these these photosynthetic mechanisms are not using water. So they are not emitting oxygen in these systems. And last but not least, we have sulfite-reducing bacteria in soil environments. So let's talk a little bit in detail about each one of these steps on the cycle. As you can see, similarly to the carbon cycle and nitrogen cycle, the sulfur cycle is overly regulated by microbes. So there are specific groups of microbes which are very important in every step of this cycle. So first of all, the assimilatory sulfate reduction, it's a pathway that for many microbes, prokaryotes, fungi, many organisms that convert the inorganic sulfate into sulfide. And then the sulfide is incorporated into carbon molecules to make amino acids. It's important to say there is an important molecule in this process, which is the adenosine phosphosulfate, and then the phosphoadenosine phosphosulfate, which is another step in this cycle. So it's very hard to reduce a sulfate molecule by itself. So there is a stepwise reduction of these molecules into these adenosine phosphosulfates and phosphoadenosine phosphosulfates, which makes it easier for the further reduction and incorporation of these sulfides into organic molecules. And then if you have these organic, these amino acids carrying sulfur, then this sulfur can be used as a process called desulfurization, which will end up emitting H2S again. There is a difference between dissimilatory and assimilatory sulfate reduction. Assimilatory, the end product that you have, is building up organic molecules containing sulfate. And dissimilatory, the objective of these microbes in the dissimilatory sulfate reduction is to use the sulfate as an electron acceptor in the respiration process. Yeah? So you see both of them are using these APS molecules in the process, but when you have the dissimilatory sulfate reduction, what's actually happening is that you're using this in the respiration process. Yeah? In the respiration process, whilst in the assimilatory, you are using it to incorporate this sulfur into amino acids, and then essential amino acids for making the proteins inside the cell. These sulfate-reducing organisms, they are not, most of the time, not exclusively using sulfate, yeah? And they can be using other sulfur molecules like intermediary molecules like thiosulfate and also elemental sulfur. But also can be using nitrates and iron and hydrogen on this anaerobic respiration process. So it could be that these microbes are very specific on the sulfur cycle, or they can be acting in many different elements, using them as electron acceptors for anaerobic respiration. So disproportionation is a very interesting process, and it's very clever at the same time. Disproportionation or dismutation, it's defined as a redox reaction where you end up with both an oxidized molecule and a reduced molecule at the same time. So what happens is when you have these anaerobic conditions, some microbes can use the sulfur both as an electron donor and an electron acceptor. So some of the sulfur will be oxidized and some of the sulfur will be reduced, yeah, in this respiration process. So it's kind of a way of, if you have an intermediary product which is not fully reduced or not fully oxidized, then you can use this intermediary product to build up both oxidized and reduced molecules in these systems. The main microbes described with these disproportionation mechanisms are the sulfovibrio and the sulfocapsa. So we have microbes, colorless aerobic bacteria that are causing sulfide oxidation processes, and these are some of the genus of microbes that are doing this. Like, for example, Biogeochemical. Yeah, Biogeochemical. They're very beautiful microbes to look on the microscope, although they are colorless. Yeah. Why do they do this? Why do they cause the sulfide oxidation? The main reason why they do this is because sulfide is very toxic to most microbes. And most microbes, they don't have tolerance to deal with excess sulfide on this system. So these microbes are oxidizing sulfide as a way of detoxifying the environment, detoxifying the sulfides from these systems. The next group of microbes, and I'm showing here a Gradski column on the right, and I will talk about Gradski column in a bit. Next group of microbes are purple sulfur photosynthetic bacteria, yeah? And I will talk about purple sulfur bacteria and green sulfur bacteria. Purple sulfur photosynthetic bacteria, they are photoautotrophic, but also photolithotrophic. So they use the sulfur as an electron donor, and they also need sunlight to process this photosynthesis. The pathways and mechanisms of this photosynthesis, they are diverse. They're different forms of structures and membrane structures that are performing this photosynthesis, and this is grossly classified by the color of the microbes, which were first described as because of this purplish color that you can see here on the photographs. So you have, these are proteobacteria, and we have two families, Chromatiaceae and Ectotioordospiraceae. Some of these microbes are not fully anaerobes, but they're microaerophilic microbes. The purple sulfur bacteria, they're very important in biotechnologies because they have a big environmental role in aqueous systems, and they are being used for bioremediation and waste treatment quite a lot. It's important to say that both purple sulfur bacteria and green sulfur bacteria, they are not using water, and therefore they're not producing oxygen on this system. In the photosynthesis, yeah? They're not producing oxygen. The sulfides or thiosulfate are the electron donors in the systems, and they produce granules of elemental sulfur. These granules can be deposited either inside the cells or outside the cells, and there's a different classification for the microbes based on this type of precipitation of elemental sulfur. And then you will have there are different membrane structures also, and these different membrane structures they also use to classify these microbes. So this is it's not a single mechanism, but is quite broad and interesting. The green sulfur bacteria, they are a little bit, they're more tolerant to hydrogen sulfide than the purple sulfur bacteria. Mainly they are from a family called Chlorobiaceae. They are obligate anaerobes, and they're photoautotrophic. Also, you can call them photolithotrophic bacteria. It becomes a little bit messy because these microbes are both phototrophs and they're chemolithotrophs at the same time, because they need the sunlight or light to move the electron transport chain in these systems. Whereas the electrons are coming directly from sulfur molecules. So these bacteria are non-motile. They are mostly stain fixed on the soil. They're not. There's actually some that are gliding, but not much. Most of them are just non-motile bacteria. As I spoke before, sulfide ions are electron donors, and they use the reverse tricarboxylic acid cycle to perform the carbon fixation. One of the interesting facts about these bacteria, they can be found very deep in water systems, even where there's very little light coming through. So they can live out of very low light concentrations. Yeah. The pigments are and the systems are bacteriophils, and the structures that perform this photosynthesis, they're called chlorosomes, chlorosomes in plasma membranes. They deposit elemental sulfur outside the cell. So to wrap up the sulfur cycle as regulated by all these different microbes, we can talk about Gradski columns. Gradski from Wiener Gradski, and Wiener Gradski is the father of soil microbiology, and he started studying the different role of different microbes in soils by creating these very diverse systems where you can have a gradient from aerobic to anaerobic conditions and a presence of different molecules and different strata of these columns. And then the type of microbes that were growing in these systems that he would look into through microscopy and study these microbes directly on their environments, not by isolating them. So the Gradski columns are columns where you will have normally a source of sulfur, a source of carbon, organic carbon. You will have also in this system iron and carbonates. And what happens is by doing this and filling this column with water, normally you start this column with mud from some water system, because these contain the inocula that you need for the growth of these microbes. And this mud column will start building up these different colored strata that you find here. I built my own Gradski columns. It's in my office in the lab, but they were perhaps too anaerobic. Yeah, you get a very high compression of this strata near the surface. So in the very bottom, you will have the most anoxic condition, and you will have, because of these highly reducing conditions, you will build up hydrogen sulfide, and this hydrogen sulfide is toxic for most organisms. Yeah, and then as you go up, you will have that you will find different strata of microbes. First, you have chlorobium or green sulfur bacteria, and after that you will have purple sulfur bacteria. Then after that you will have purple non-sulfur bacteria. Then you have iron oxidizing bacteria, and then in the top you will have aerobic microbes, heterotrophic aerobic microbes, and cyanobacteria will be creating the oxic conditions on the top, and these heterotrophic microbes will be the one fully oxidizing this sulfur back into sulfate. So there is a gradient of sulfate. Sulfate is diffusing down, hydrogen sulfide is diffusing up and being detoxified as it goes up. And the tolerance to hydrogen sulfide is conditioning which microbes can live in which strata at the same time. So these are very interesting systems, and you can encapsulate, by doing this, all these different groups of microbes in a bottle, and you have the full sulfur cycle in a bottle, and you can see also interaction with different other nutrients, for example, iron, carbonates here, and nitrates also, and you can observe this with a very nice didactic way, which it shows in different colors of microbes. So it's beautiful to see, and it's very emblematic because these were first built by the father of soil microbiology. So when we look in agronomic systems, when we're thinking about plants, why do we think sulfur is so important? Why do we care so much about sulfur in the agroecosystems? The main reason is sulfur is an essential nutrient. So sulfur is an essential nutrient for plants, and it is essential because it's you cannot replace it by any other nutrient, and it's part of very important structural molecules like amino acids, like cysteine and methionine. It's involved in many processes like the conversion of nitrate into amino acids and then into proteins. It's important on the stability of the proteins because the proteins, when they fold, they can form this, in eukaryotes, they form these disulfide bonds. Sulfur is part of vitamins. It's important for chloroplast growth and function and photosynthesis, therefore. Sulfur is important in nitrogen fixation by legumes. And these are some of the reasons why sulfur is so important for plants. So normally in soils you will have that sulfur will be deficient, and this is what makes sulfur a macronutrient so relevant, because especially these days when you don't have atmospheric nitrogen deposition so much, you will have many when you have low organic matter, and you will have in sandy soils. These are the conditions where you have higher deficiency. When you have sulfur deficiency, what you will see is this chlorotic yellowing of the plants, which is mainly due to the sulfur being fairly immobile in the system. It's not well recycled, so you cannot see too much patterns in these leaves. It's just a yellowing of the leaves. It's the main symptom of deficiency. It decreases plant growth and decreases the yields of crops. So of course you want to add sulfur as a fertilizer. Yeah, continuously sulfur is becoming more and more limiting in soils because you have less atmospheric deposition. The main sources that you have from the sulfur as a fertilizers are mined elemental sulfur that you can use to make sulfates, or you have elemental sulfur, which is our byproducts of gas production also. So most of the sulfur in the soil is tying into organic matter, and the ways that you can deal with this sulfur deficiency is just by using sulfur-based fertilizer. The main sulfur-based fertilizer in general is gypsum, which is calcium sulfate. Yeah, but this is it's used in alkaline soils for mitigating salinity issues. Gypsum will help to leach sodium salts by replacing them with calcium. So normally you will have that soils deficient in sulfur will be light textured soils, which are flooded. The irrigation systems are doing flooding. Then you will have more reducing conditions. You will have more leaching. For example, like in rice systems, when you have very low organic matter in general, you will expect sulfur deficiency because most of the sulfur in soils, they are tied to organic matter. You can also have sulfur toxicity symptoms, but this is normally caused by hydrogen sulfide, not by sulfur. And some of the agrochemicals that we use, fungicides, they contain elemental sulfur, and if you overuse them, you might have that you can cause sulfur toxicity symptoms on the plants, which are burning of the leaves mainly. These are some of the sources of sulfur in soil. Gypsum is one of the main ones used, but you also have monoammonium sulfate, potassium sulfate. You have magnesium sulfate, zinc sulfate, and elemental sulfur also. So elemental sulfur is one of the amendments that I'm studying. One of the reasons I'm studying this amendment is because it is causing acidification of soils. The oxidation of elemental sulfur will render a sulfate molecule, which in the soil is in the water soil solution will be in the form of a sulfuric acid, and of course it will be acidifying the soil. Because we have highly alkaline soils in Oman, then it's very interesting that we have some acidification of these soils. This process is completely biological. The main microbes that are doing this are Thiobacillus and Acidification. Yeah, so the other thing that we find interesting is by producing protons in the system and acidifying the soil. The protons are the ions with highest affinity for the solid phase, and therefore it would replace other ions in the solid phase, like for example sodium, and it will help the sodium leach through the system with the sulfate. At the same time, if you have acidification of the system, you will have the dissolution of carbonates, and this dissolution of carbonates will release calcium in the system, and the calcium can then displace the sodium in the solid phase and help the sodium leach. So elemental sulfur will be beneficial in every time you have sodicity problems in soils. Elemental sulfur is also very beneficial, and it's a good substitute for gypsum. You need less amount, and it will be more effective because you have both acidification of the soils and the release of calcium from calcium carbonates. This elemental sulfur is also very highly convenient for Oman because during the gas production you will have that elemental sulfur is a byproduct of the processing of natural gas. And you can build up high amounts of this, and at the same time, if we need it for agriculture because of salinity issues that are widespread in Oman, then you can may give a use for this byproduct as an amendment, as a fertilizer, and at the same time, you can help deal with a predominant issue for agriculture in Oman, which is soil salinity. So this is all I had for you guys today, and this is all for the sulfur cycle. See you next lecture.